Welcome back to Monkeys on Typewriters on YouTube. In this video, we're continuing with the series on how does this end. In the previous video, which will be linked in the description below, we heard about plagues caused by bacterial pathogens, and we're following that up with some discussion about viral pathogens, including influenza and smallpox. And again, the main point here isn't the wealth of details about those particular pandemics and plagues, but the general themes, and which will then be applied to where we are with COVID and where we think things might be going. So with that, I'll hand it over to you, Arijit, and let's continue with this very interesting discussion.
Thanks, Ryan. So we're going to jump into influenza now. And influenza is a topic that unfortunately is on everyone's mind right now. When we actually put this presentation together, this was a little while back, so it wasn't really a pending news item, but it is now. And by the way, a lot of what's discussed here can also be read about in an article that Ryan and I co-wrote with Lynn Filethro called "Are We Ready for the Next Pandemic?" And really talking about the 1918 pandemic in relation to where we are today, and that you can find that on Scientific American if you want to go looking for it. So this particular talk really will now focus on this disease. And so many of you already know this. Influenza is an airborne disease caused by influenza viruses. There's a wide range of symptoms, and seasonal endemic flu is nasty. Those of you who've had it know this well, but it's not usually life-threatening. There are 250,000 deaths worldwide out of 5 million severe cases and a billion infections. So it's a relatively low death rate, but obviously that relatively low death rate, when added up throughout the world, can add up to quite a few actual deaths. So the influenza virus is a little bit different from other pathogens we'll be looking at, because influenza virus has two antigenic determinants. The hemagglutinin protein and the neuraminidase protein. These are two different proteins on the surface, and they're very much capable of mixing and matching, which we'll get into. This mix and match nature of the influenza virus really gives it quite a bit of ability to evade the immune system. Nowhere near as much as COVID does. Influenza, we think of influenza as evolving very rapidly. In context, influenza evolves thousands of times slower, even though it mutates at similar rates. And the difference between evolutionary rate and mutation rate is a significant one that, by the way, a large chunk of the scientific community missed in 2020. And many of you who follow me know this, but my collaborators and I called this out in 2020 in a paper where we showed that SARS-CoV-2 was likely to evolve very rapidly, despite not having a terribly high mutation rate, because it was very flat in terms of its evolutionary landscape, meaning that it was very resistant to mutations, or very able to work its way around mutation in the spike protein that would lead it to evade the immune system. Such mutations we found at the time in 2020 were not likely to abolish binding to ACE2. So that dance between the immune system and the virus really comes down to what happens on the outer coating of the virus, on the proteins that are the antigenic determinants. So coming back to influenza, those antigenic determinants are neuraminidase and hemagglutinin. And influenza uses a bit of a different strategy to be able to pull off that immune evasion trick. So there have been 4 major pandemics caused by influenza in sort of recent recorded history. The so-called Spanish flu in 1918, which really should have been called the first American flu. Asian flu in 1957, the Hong Kong flu in 68, and then a second H1N1 pandemic caused by a different strain of the virus in 2009. So
I'll just
when we
interject
look,
real quick. Just you
yeah,
maybe
sure,
going to
jump
cover
in.
this, but you mentioned the surface antigens, and I may be pointing out that the H and the N, when you hear all these strain names, this is what you're talking about, right?
Oh, yeah, good point. So H means hemagglutinin, N means neuraminidase. So these are the actual variations of the H and the N protein. And so what influenza does is it mixes and matches those. So H1N1 is a pair of antigens, H1 and N1, that determines the immune response to influenza. And H2N2, which came out in 1957, burst on the scene and caused the pandemic immediately. H3N2 burst on the scene and caused the pandemic. So every time you see a situation where you have a pandemic with influenza, it's not the seasonal virus that is causing the pandemic. It's a brand new virus, so to speak, with distinctly different antigenic determinants. Often the virus will have other changes as well, and we'll get into that.
Right.
But the hallmark of the pandemic causing viruses, very similar to what we saw in the last episode with plague, is they're the product of punctuated equilibrium. And so the way punctuated equilibrium works for influenza is that there are multiple reservoirs. So you have chickens, pigs, cows, all of which can get infected with influenza. And you have this ping pong zoonosis where humans will infect these animals. The animals then breed the virus within them in large outbreaks. And then those viruses come back to humans. And when they do that, you have this process, as we discussed in the previous episode, called peripatric speciation. I keep wanting to say peripatetic, but that means something different. So peripatric speciation is when you have a second smaller reservoir, second distinct reservoir of where evolution is taking place, a second niche where evolution is taking place. And then when that particular organism reinvades the original territory of that it occupied, it comes back looking very different. So you have essentially a punctuated equilibrium event where it looks like you suddenly are seeing a huge amount of evolution in the blink of an eye. That's not what's happening. What's happening instead is that evolution was happening out of sight. Now, we've discussed that this actually happens with COVID as well in long-term infections, where that second reservoir is not a different species necessarily, although COVID does infect other species. The major driver of COVID evolution is that second reservoir is long-term infections. So in the case of influenza, we don't know when influenza emerged into humans, but influenza has a segmented genome, which means it has little sort of cassettes, if you will, of genes that can sort of move around together. And that segmentation with certain recombination sites within the genome being favored for these swaps allows recombination to drive evolution very efficiently. Particularly, recombination between the H and N genes are the ones that drive that antigenic shift. And the novel HN antigens then drive new pandemics. So the exact determinants of virulence, whether they're in the HN genes or whether they're outside the HN genes, is poorly understood. It's very likely that what happens in these pandemic influenza strains is not just that you have changes in the H and N genes, which will give you changes to how well the immune system responds, and therefore some degree of loss of protective immunity and some degree of increase in the rate of fatality from that alone. But in addition to that, there are other changes that happen in the viruses. The changes that we see on the surface are reflective of the fact that this virus has had time to evolve somewhere else. So confusingly, the virology community has two different names for this. Antigenic drift is a set of small mutations that accumulate over time. And antigenic drift is what you see from season to season with influenza. An antigenic shift is when you have this kind of punctuated equilibrium event happening from peripatric speciation. So antigenic shifts are big jumps in antigenic changes. Antigenic drift is confusing as a term because you can easily confuse it with drift in evolution, which simply means changes in sequences that are not being driven by natural selection. But that's not what it means here. It just means little changes versus big changes. So in any case, the point being that people have known for a long time that influenza pandemics are caused by antigenic shifts. And these antigenic shifts are essentially large changes in the virus that result from evolving for a period of time in other species. Now, as you know, this separately from COVID, this also has implications for where we are with the current situation with bird flu. Because what's happening with bird flu is essentially you've had these large outbreaks that are running free and running wild in these populations of livestock. There's actually reporting has been suppressed. There's no tracking going on. We don't know exactly what's going on. And that many people have pointed out that that's a ticking time bomb. In the article that Ryan, Lynn, and I wrote together, we point out that if we had another outbreak of another pandemic level threat emerging into the population from the mishandling of bird flu that is currently sort of the MO, we're not much better off than we were in 1918 to deal with that. Medical science hasn't evolved any further, really, when it comes to the major killers of influenza. And we're kind of just as defenseless and just as vulnerable, with the one major difference being that in 1918, it would take a week to get from London to New York, and now it takes a few hours. So we're in a much more connected world. So do dig up that article if you get a chance. But digging into the 1918 pandemic next, let's talk about sort of how this plays out, looking at one example of an emergence event that was driven by punctuated equilibrium. So in 1918, soldiers at Camp Funston, Kansas, who showed up with symptoms of a severe respiratory illness, started falling sick and actually dying very quickly. And there was sort of a mild first wave. There were some deaths, but it wasn't really sort of devastating. And then it looked like it had passed. The problem was solved, so to speak. But in fact, a second wave occurred several months later with a much higher fatality rate. And then there was a far less deadly, but much longer third wave that lasted all of 1919 and into 1920. So a total of 50 to 100 million people died worldwide. And adjusting for changes in world population, if we had had that pandemic happen today, we would be looking at a death toll about 4 times higher, so about 400 million people, which would eclipse where we are with COVID to date. But the main point here is that the pandemic happened in different waves, and those waves actually had not necessarily a monotone decreasing rate of death, but sort of the death rate was jumping around. So why was that? There's a lot of questions here. Why was the death rate higher in the second wave? How did the virus actually disappear? And so, as they say, inquiring minds want to know. So the first question to look at is, where did that virus come from? So the H1N1 virus may have been circulating in pigs as early as the turn of the 20th century. So what these guys did in this particular paper was they went back and now what's happened with influenza, which is another fascinating story in itself, and people have actually written books about this. They had people go out there and dig up viruses from the permafrost up in Alaska of victims of the 1918 flu. And they sequenced the actual 1918 virus from those individuals. And in fact, the guy, I'm now blanking on his name, but the guy responsible for that in one of the sort of, broken clock is right twice a day kind of situations is responsible now for the NIAID, which is really cool. But this was a very interesting piece of work. And so what they did was they dug those viruses up, and then what they went and did one step further was they sequenced them. Once they sequenced them, they were able to begin to put a patchwork together of sort of what are the different, where does this fit in the evolutionary tree of influenza viruses? So what they were able to find was that when they looked at it in comparison to previously circulating swine flu strains, as well as mammalian viruses, they were able to find that essentially the portion of the genome that was human H1N1, specifically related to 1918, not the seasonal H1N1, which exists, probably dated to somewhere between 1910 and 1918. And that portion of the genome was in the HA gene. So essentially, when you looked at these changes, there were multiple different genes that sort of showed up all of a sudden in the BM is the alternative name for that virus, a Breivik mission, which was where they dug it up in Alaska. So the BM 1918 H1N1 virus had acquired, at a minimum, the hemagglutinin gene, but also a number of other genes right around the time that the pandemic kicked in, which was sort of fascinating, which meant, yes, the acquisition of that H gene was obviously a driver in the pandemic. That's what makes it different from seasonal H1N1. But the other piece of it is that wasn't the only thing that changed. In the H gene, a single nucleotide change was sufficient to make this swine flu hemagglutinin able to infect humans effectively. And so influenza viruses that are adapted to birds or are they preferentially bind sialic acid receptors with alpha 2 3 linked sugars. And human adapted influenza viruses preferentially bind receptors with alpha 2 6 linkage. And that switch requires only one amino acid change. So there are 5 samples of the 1918 virus, and all of them have this single amino acid change, suggesting that that was a major driver. Um, so that was the change in host transmissibility for humans. But that wasn't the only thing that was going on in that virus. So people looked at the 1918 pandemic virus and compared it to contemporary human H1N1 virus, which, by the way, is not a great study to be doing. Like, ideally, people shouldn't be doing this stuff like this. There are better ways to do this. But what these guys did was they took the 1918 pandemic virus and they infected non-human primates. Again, so, you know, playing with fire here, given what we know today. Like, I think when people do experiments like this, they're like, oh, it'll be fine. It really won't be fine. We don't have the medical infrastructure to deal with, a 1918 pandemic influenza virus lab leak. And in fact, the 1960s influenza pandemic almost certainly was the result of a lab leak. So this is not the greatest experiment to have done. But what they did do was they showed that this was the 1918 pandemic virus was highly pathogenic for non-human primates. Okay. Now, what they did was they were able to show that when they took this virus and they looked at replicative ability, what they found was that the virus was much more able to replicate efficiently in upper and lower respiratory tracts, whereas the modern H1N1 doesn't do so well in the lower respiratory tract, which would be, one potential reason why. And what they found was that this was more related to the N gene. So H1N1 genetic constitution made a difference. Now, at the same time, there was an pathogenic determinant of virulence coming from the hemagglutinin gene as well. What these guys showed again in a third, cursed experiment was they infected mice with it, and they showed that basically if they just took the HA gene derived from the 1918 virus, that alone was lethal to mice, while contemporary seasonal influenza was not. So it pinpoints at least some of the determinants. What this shows is that, in fact, it's not that there were other environmental factors involved. The H1N1 1918 version was in fact more deadly, even by today's controlled conditions, and that you could pinpoint that both to the raminidase gene and the HA gene. And so there's a lot more in this paper, and it's sort of, a grimly interesting paper to look at from that perspective. Hopefully, there's not a pressing need for a lot more studies of this type. But the point is, the H1N1 gene was not only more transmissible, the H1N1 from 1918 was not only more transmissible, but was also associated with increased virulence. Now, what's interesting, though, is, as we discussed, the death tolls in the different waves were quite different. So this is the first wave, as we discussed in the summer, that was relatively mild. And then you can see the death toll in the fall was higher. And then in 1919, you had essentially a much longer wave that had lower overall mortality. But so these three pieces of it were really open questions. One is, where did it come from? Why was it so much deadlier? And now we have answered some of that. And the answer is because it was an emergence event driven from livestock. We know it was more deadly because it had harbored multiple genetic changes relative to the seasonal pandemic flu seasonal influenza H1N1 that we're familiar with today. And then we also know that it had a number of changes that could be directly pinned both to increase transmissibility and to increase virulence. So why was the second wave deadlier, though? Well, it turns out that during this particular pandemic, there was very little viral evolution going on, which is sort of surprising, right? Because we talked last time about the Black Death, and we talked about how it was almost as if time had speeded up for the Black Death, Yersinia pestis pathogen, during the Black Death and during the second plague pandemic overall. In the case of influenza, through the 1918 pandemic, there was actually very, very few changes in the genome. So influenza just behaves differently. And so when people sequenced a bunch of viruses that they were able to find through 1918, 1919, and put them on a tree, they all sort of clustered together pretty neatly by wave. So the autumn 1918 wave clusters neatly. Um, some summer 1918 viruses cluster with the autumn months. And then there's this other clade, which is really going into winter and spring of 1918, 1919. And so that was a different clade. When they traced these phylogenetic trees back using certain assumptions about the rate of change, they can they can see when it diverged. So this particular clade rooted back to the original virus, the other clade, before the pandemic hit. So these were actually different clades that emerged more than once from livestock, which is kind of crazy if you think about it, right? It's like it's both extremely plausible and also sort of how this works. We've already seen this happening with bird flu, where you had um, a number of clades enter humans already, and they've not been very transmissible. But you're seeing those sort of animal to human transmission events happening on multiple occasions. So what this shows is that the second clade that showed up was actually deadlier and was just genetically distinct from the first one. So then that begs the question, okay, so that's what happened with the second wave, but how did it then attenuate? How did it then become milder, right? It's a legitimate question because we know that seasonal H1N1 is not as deadly as 1918's H1N1 was. And in the lazy telling of the story, you'll often hear that the pandemic influenza was sort of extremely virulent, but after a while it sort of lost its potency. Well, actually, that's not what happened. Um, So people have again done phylogenetic sort of trees, and they've taken a wide variety of different influenza strains from swine flu, avian flu, and humans over the years. And they've sort of constructed this trees like this. And so the year is listed over here. France 88 means isolated from France in 1988, not surprisingly. And so when they root these things back, what they find is that the seasonal H1N1, so this is actually right here is 1918. So the question is, where does 1918 fit with the other seasonal H1N1s? And the answer is you have to go all the way back to the root of the tree. So what this means is that this clade went extinct after 1918, because you don't see other examples of that. And these other clades were derived from a different emergence event coming out of swine. Just in the same way you had two waves coming out of swine in 1918, you had other waves come out that created the seasonal influenza H1N1. But so that's pretty striking. If you think about that, what that means is that you weren't looking at attenuation necessarily. You were simply looking at the fact that the scary version of the virus, which by the way almost certainly caused, and we'll talk about that, soon, but it caused human behavioral changes, didn't do as well. It wasn't because it was less transmissible. It was pretty darn transmissible, but it didn't do as well in practical terms. So it went extinct, while other H1N1s that were much less scary have continued to thrive in the human population. So that's your point of convergence here between the seasonal on the top and the 1918 flu in the middle. Here's a second example from a different paper that makes the same point. And what you're looking at over here is seasonal H1N1 versus 1918 H1N1. And when you compare them, each individual gene was derived from swine flu at different points. So you can see that the composition is kind of, although there are some shared genes between the two, particularly for hemagglutinin and neuraminidase, the dates for the seasonal H1N1 are just different. So again, the composition of the H1N1 virus in the seasonal form and the pandemic form just look different. Now, I will say, however, that this is somewhat controversial because there has been some debate on this, and one of the sort of slightly technical things is that there's an assumption about the rate at which the virus evolves that goes into this. It's called a Bayesian molecular clock. And that does drive the answer somewhat. So in this particular case, these authors showed that you could place the two in distinct arms relative to swine flu, depending on when you on what assumptions you make about the molecular clock. So it's, there's some degree of emergence, if you will, no pun intended, about sort of the picture when it comes to this. But if you look in both of these, the seasonal 1918 pandemics are on different branches, whether they branched away from each other or whether 1918 branched away from swine flu after seasonal peeled off is what's being debated in this particular figure. But I'm just pointing this out to show you that there is some degree of slop in terms of how these things show up. But in the big picture, whether it's the picture on the left or the picture on the right, uh, I think everyone agrees that the 1918 pandemic was a different clade and it went extinct. Why did it go extinct? Well, it turns out that influenza is less transmissible than other diseases. And in the case of influenza, um, there was this was 120 years ago. There was a very strong pressure to wear masks. So, in fact, it was simply wearing gauze masks, just like, you know, straightforward, almost like cotton, cotton cloth, nothing fancy. And what they ended up doing was they ended up hindering the transmission of the virus just enough. Between public health measures and herd immunity, the virus just didn't have enough to work with. So that's an actual picture from the pandemic with the nurses and doctors wearing masks. It's just shocking to see actual medical health professionals wearing masks in a pandemic. That's brand new to those of us who have lived through this one. Um, People have actually done epidemiological modeling to show the impact of public health measures during influenza. And what they were able to show over here, what they did is they fitted an epidemiological curve, and they were able to do the counterfactual. They were able to say, well, what would have happened in each of these cities if public health measures were not in place? So the blue crosses are the actual deaths. The red is the actual curve, the epidemiological curve. The bright green lines were when public health measures were implemented. And what you can see is that the public health measures being implemented were kind of concurrent with just before the wave or concurrent with the wave. So they're right around that time. And the dark green line is what would have happened if those public health measures had not been in place. So you can see, depending on the city, and the effectiveness varied from city to city, but pretty much every single time, the public health measures had an impact on the final deaths. So again, these were widespread public health measures, and they had a substantial impact on the outcome. There's studies that show similarly for COVID that the public health measures during the early part of the pandemic were impactful, and they did reduce the total number of deaths to varying degrees, but they did reduce them. So this is not surprising. But what's interesting is that for 1918-1919 influenza, there was much more public support for it. And in fact, people would get thrown in jail for not masking and so on. And so that sort of public support for it, and plus, given the fact that this was a different virus and easier to defeat with public health measures, meant that the public health impact on this virus was much larger than what we've seen with COVID, in a positive direction. I mean, if we look at it, if we're direction agnostic, public health has played a big role with COVID. But we can come back to this Red Queen's race picture. And so with 1918 influenza, this is what happened. We had emergence, we had the establishment in the population. Then we had mass mortality with very limited evolution because we had almost no wave upon wave iterative sort of circling back and hitting the population, followed by extinction. So this almost is a best case scenario, right? And what's shocking is that all of our pandemic planning prior to 2020 was based on influenza. And so if you look at it, we were really planning for the best case scenario. We were planning for a situation where all you needed to do was simply lock down for a few months and it would be fine, which is kind of what happened in influenza. They didn't account for the fact that other viruses didn't behave that way. Simply defeating the first wave was not going to be enough. There was just no gas left in the tank after the summer of 2020. And this is why, because we were planning to the best case, which was the 1918-1919 pandemic. So, in fact, indexing on 1918-1919 exaggerated the impact of the public health measures that they were planning to put in place. Just masking and locking down was never going to be enough for COVID. And there was no plan B. There was no actual impact on sort of bringing up a portfolio of antivirals or bringing up other kinds of prophylaxis measures, cleaning the air. None of that came up because the idea was you just had to duck it the one time and duck the wave and you would be fine. Um, and and sort of unfortunately, COVID didn't play out this way. So there are a lot of retrospectives about lessons from the Spanish flu. This writer Harold Brusso talks about it in a paper from a few years ago. I think both you and I were quoted along with him at some point with Aaron Prater in Fortune, Ryan. He's listed along with us in one of the articles that was written. But but Brusso's examples or Brusso's lessons from here, I'm going to contrast with what we actually know today. So his point is, look, typical pandemic will be about 2 to 3 years. So even after the pandemic phase is over, SARS-CoV-2 will kind of taper out after about a decade and we'll have winter epidemics. And then, his point is that viruses cannot easily reacquire the virulence of the initial trans-species infection event. And so, as it so happens, some of this is very widespread, right? These are very widespread beliefs that novelty is severity, for example. So widespread belief. It does so happen that pandemic viruses of high consequence are both highly virulent and novel. That's true. But there are plenty of examples where highly novel pandemic viruses have had low consequence. For example, the 2009 H1N1 had much less impact than 1918. So it's not immediately the case that it's going to burst on the scene extremely virulent. It's not necessarily the case that that virulence will taper off. All of those things are sort of up for negotiation. It is true that when a brand new virus with new antigenic determinants comes into the human population, there is a certain degree of protection that prior exposure to that particular virus across its different strains, provided that is being lost. So there's an increased virulence component from that. But whether that increased virulence component, increased sort of realized virulence component, if you will, whether that alone is enough to cause a disastrous outcome it's not necessarily the case, because if you have a loss of sort of intrinsic immune protection due to the adaptive immune system having seen that general class of virus before, it doesn't necessarily mean that the virus itself is extremely pathogenic and can kill that efficiently. For example, 1918 H1N1 was able to efficiently infect the lower respiratory tract. Combining that with a massive loss of protection can have much worse outcomes than if it was simply an upper respiratory tract virus that was also immune evasive. So there's two components to virulence there. So with all of that said, let's compare and contrast this, keeping in mind that this is inductive reasoning with an N of 1, right? So the myth is that pandemic lasts 2 to 3 years. As we talked about in an earlier episode, there's no set timeline. The Antonine Plague lasted for decades. Black Death lasted for 7 years. Second plague pandemic lasted for 2 centuries. Your mileage may vary when it comes to a pandemic. There's no contract anywhere that says it has to come to a close up at the 3 year point. The virus attenuates at this point. Attenuation, as we talked about in the first part of this, in the very first episode, attenuation is a straight up myth. We're in the Red Queen's race. Sometimes it will attenuate, sometimes it'll get worse. It didn't happen in 1918. It hasn't happened in other pandemics. The Delta wave was much more deadly than the 2020 wave. So even with COVID, it hasn't happened. Settling into a predictable endemic state, again, not guaranteed. You could have a different clade that is responsible for the seasonal version of the virus. And waves in the winter, again, that's not a general thing. The inference that somehow because it was happening with seasonal influenza, it was going to happen with seasonal COVID, that was another myth.
This is another one of those ones where slides having been prepared a while ago, we can now even update and say that at the time of recording, we just had a winter with a very relatively low rate of COVID infection because we had a fairly significant wave in the previous summer and fall. So
yeah,
it's
that's a great
clearly
point.
not. I mean, even
It's
now, we
clearly
can
not
say
seasonal.
for
Yeah,
sure it's not settled into a
a
seasonal
seasonal.
winter respiratory virus cycle
Yeah,
at all. It has a lot more to do with which variants have arisen when, levels of
yeah. Viral
community
evolution sets the tempo.
maintaining. Yeah.
Yeah. And COVID will never be seasonal because viral evolution is what's setting the tempo. Exactly. So all pandemics are not the same, right? So 1918, 1919 was the best case scenario. These are self-limiting pandemics with no red queen dynamics in play. And so in these cases, you can flatten the curve. And then hopefully, if we don't have sort of a very large sort of suicide at the hands of infectious disease crowd, then the pathogen will eventually go extinct. Right. So that's your best case scenario. Again, that requires a virus that's not, or pathogen that's not that transmissible. It requires human beings to take it seriously and to be scared with the mass mortality event, which human beings have a way of being scared when mass mortality kicks in, by the way. So all of that is fine. Again, that's one way a pandemic can play out. But that's not the only way. We saw with the plague, it didn't play out that way. What you had was emergence, you had establishment of the plague, and then you had repeated waves with mass mortality and rapid evolution until people decided not to live with the plague. So these are two very different scenarios, with and without the Red Queen. And so understanding whether you're dealing with a pandemic that has Red Queen dynamics or not is really, really important because otherwise you're just planning for the best case scenario. The pandemic scenario on the top is the only one for which flattening the curve sort of makes sense. In the in the bottom case, flattening the curve is meaningless. There's no such thing. And really, you're constantly running that treadmill until you decide to get off it. Um, and so that happened with the plague, that's happening with COVID. So this is why when people talk about 1918 influenza in the context of COVID, which is, by the way, most of the poorly thought through discourse on the damn thing for 5 years now, I always roll my eyes either internally or externally because it's just such a bad sort of thing to index on when it comes to a Red Queen pandemic. So coming to the take homes for influenza. So punctuated equilibrium, which is reemergence from a disease reservoir, represents a pathway to pandemic events. And you really need to surveil the disease reservoirs and make predictive sort of efforts when it comes to monitoring threats. Now, we actually do that for influenza, or we used to do that for influenza until people started walking around the place with chainsaws, metaphorically speaking. But that's a really important piece of infrastructure. We don't have that for COVID. The other thing to take away from influenza is that public health interventions can make a huge difference in the early stages of a pandemic. And when you have a Red Queen's race in motion, flattening the curve will not work as well as it did for influenza. So that wraps up the influenza portion of it. We only have about 10 minutes left. So I suspect that this, again, will this series ever end is my question now. But I suspect we may have to punt the smallpox to the next hour as well.
Fair enough. Fair enough. There's lots to say.
Yeah, there's a lot to discuss even for this one.
Yeah, and I think some of the general concepts that are, I hope, emerging and becoming clear through all of this are, we don't really, there is no automatic pattern followed by any particular pandemic or plague, and the idea that you just need to wait it out and eventually it will attenuate and then you're fine is not how it has worked historically. It's always involved some manner of significant intervention to get it under control, or a very, very large amount of human death and suffering, right? So
Yeah,
you could take an evolutionary strategy, speaking of human evolution in this case, but that comes at an enormous cost. I think one thing too that people, I hope, lessons to be taken from influenza, which you've touched on here as well, but is the idea that you've got a regular source of reemergence or new novel emergence, let's call it, new clades evolving in a non-human reservoir and then entering the human population. So the idea that you just have to deal with the one that is currently in humans and take no steps to mitigate future transmission from non-humans to humans is very problematic. And I think that we sort of see that with coronaviruses too, which is there's lots of them out there, and there's always the potential for another one to make the jump from whatever host species it is into humans. And even if humans are essentially an evolutionary dead end for that lineage of viruses, it will still wreak havoc while it's here, and then
right,
there could be another one come along. So
right.
there's just really missing the sort of much bigger picture besides humans or besides the current wave or besides, we tend to get very myopic right now, and anything we do assume about the future is based on a myth that if you just wait this out it'll be fine, and that none of that is actually accurate.
You make a great point. I mean, if you look at it, um, it is almost certain that there was an out of control pandemic in livestock at the beginning of the 20th century. That livestock pandemic was the gift that kept on giving. It sort of spread two rounds of pandemic influenza and one round of seasonal influenza, brand new clade H1N1, into the population that didn't exist before that,
Yeah.
which is kind of wild when you think of it. So I think the idea that somehow these kinds of livestock pandemics can just be swept under the rug is also very dangerous because livestock is, livestock represent the best shot of getting something into humans from another species because most of us don't farm bats for food. We do farm other animals for food. Which animals are capable of harboring viruses that infect us? So all the talk about eating bats and pangolins, which is tinged with a little bit of, I'm just going to put it out there, sort of questionable ethnic assumptions.
Mhm.
Um, I think that's all great, but the point is, we actually have the bad habit of eating large numbers of animals in that are farmed in very dense conditions that would make a bat cave look suburban in comparison. And we have no way of apparently either monitoring or controlling infections in these spaces, or so we've been told. So the idea that somehow, these kinds of livestock pandemics are just benign events that, epiphenomena that only impact the price of eggs, is something that we seem to have settled on now. But I don't think that's going to be how it plays out.
No, there's a very clear willingness to, vilify wet markets or transfer of wild caught species into the food chain or what have you. But yeah, I
Yeah.
would argue
I
that
mean, how many
high
pangolins get
density
eaten versus how many
captive farming.
chickens, right?
Exactly. You've got
Right.
literally billions of livestock kept in very close quarters with regular contact with humans. So
Right.
and I think there tends to be you've talked about it before around, the sort of turkey fallacy, right? Where because we haven't seen a massive pandemic emerge from those livestock populations yet, it isn't going to happen, right? That it's been false alarms.
From those livestock populations in a century.
Exactly. And moreover, the conditions now, and we've said this before, that the conditions today are significantly more amenable to a massive pandemic spread than they were in 1918 with global travel, with larger population densities, with
Yeah.
factory farming. I mean, it's worse, not better than it was.
Right. And as we discussed in our Scientific American, the ways in which influenza kills, which is acute respiratory distress syndrome, secondary pneumonia, um, these kinds of things, on cytokine storm, these three killers, if you will, for pneumonia, for influenza, which by the way were also the killers for COVID in the early stages of COVID,
Mhm.
uh, we haven't done that much better with them. Like cytokine storm has a very low survival rate today. ARDS has a very low survival rate. And secondary bacterial pneumonia in patients in the ICU has a shockingly low survival rate. It's like, I think 40 or 50% of people with that die. So we're not any better off. We have a we have a medical system stretched almost to breaking point. We have no surveillance mechanisms in place for this, for H3N2. So in in many respects, except for the hubris, we're no different than the people a century ago.
Yeah. So on that on that note, I'll invite everyone to check out some links in the description. I'll provide a link to the Scientific American article that we mentioned, a few other things, as well as other video series and the first parts of this series. And we hope you'll join us again for the follow-up, where we will get to smallpox and then eventually to COVID and other current risks and where we are with all of that and how those may end. So thanks again, Arjit, for a really very informative and interesting presentation. And hopefully we'll see everyone again soon.
Thanks, Ryan. See you again soon.